Good. Hello, everybody. I will go to my screen for sharing. Are we good with seeing the first page of the
Yep, looks good,
slideshow?
Bill.
Okay, very good. Hello, everybody. ASCE, EWRI. I like this group, a lot of good engineers. We'll be giving the presentation today on the Bioretention Hydrologic Performance study, and it's the third in a series of 3. Jenny Saltonstall from Associated Earth Sciences will be with me today to co-present. We have Anna Maria Clark and Chris Wright from Raedeke Associates. They're not on today, and Doug Byerline, Clear Creek Solutions, that was working on the hydrologic design assessment portion. And the Raedeke Associates folks were the vegetation specialists. I wanted to recognize our sponsor, City of Olympia, and our Department of Ecology project manager, Chelsea Morris. And this work is funded by the SAM Stormwater Action Monitoring Program that is funded through the NPDES program. And let me see if I can enable my down arrow. There we go. This study is in the third of 3, and the focus of this one is, how are they aging? How are bioretention facilities aging? We looked at 50 bioretention cells 10 years or older all throughout the Puget Sound area. And we characterized the plant community, measured the cell infiltration rate and soil composition, and also assessed the owner's operations and maintenance activity, and asked the question, does the current infiltration rate in plant community, how does it compare to the initial design and plant community? And what do we gain from that in design recommendations? Our 50 sites spanned about 26, as I recall, jurisdictions. A number of them had multiple cells that were included in the study, 25, 26 jurisdictions. And they spanned all the kind of climatological and geotechnical conditions throughout the Puget Sound area. We'll start with the vegetation results. And by way of explanation, I understand the audience here today is largely engineers, so we'll do a little explanation on the wetland indicator status, and that is abbreviation WIS, and it's how likely a plant species is to occur in wet versus dry soils. So this, it uses the word wetland because this wetland indicator status classification is part of the formal wetland delineation process, but that's simply for classifying the plants. And we use that as a way to, again, recognize whether the plant is a little bit more tending to be in wetter conditions. And when we say wetter conditions, it's really the subsurface soil conditions in the lower part of the plant's root zone. And often for very wetland species, the water sits for a long time. In wetland conditions, the water sits for a long time and actually becomes anoxic, and there can be associated conditions in those wetland conditions that wetland associated plants are adapted to. Bioretention facilities are not like that. They do not accumulate water for long durations of time to be classified as a wetland. So remember, bioretention facilities are not wetlands. They're actually very well draining facilities, and you'll see some of the results of that, the indications of that, with both the plant species that occur and the infiltration and soil texture conditions that Jenny will talk about. We'll go through a number of example sites just to give a visual and plant condition result that we see for a number of these sites to start the discussion. This is one site up in Arlington Boulevard, obviously in a right-of-way, heavily maintained, probably has some foot traffic in it, and no irrigation. So to summarize what we found in these facilities for plant condition, and we did a transect along each of the 50 sites, and the transect had quadrats along the station where our botanists identified the plants in random quadrats throughout the length of the facility. And the wetland indicator status then was assigned to each of the plants that we saw. And we also got the original planting plan for most of the facilities and assigned the wetland indicator status for those plants. And we lumped them together to compare the original planting plan with what we saw presently. And there are actually 5 categories of WIS, and we lumped together the wet end, which is percent on the left, the wetter plants, percent OBL, which is obligate wetland, and facultative wet. And in the middle, the classification is percent facultative, which means it's adaptable in both directions. And in the drier end, it's the FAC-U, which means facultative upland and upland, and those are very dry plants. And what we saw in many of these sites was a shift from a wetter list of plants that were planted originally, that's the blue bar, with a shift over 10 years to a drier set of plants, the orange bars. This is a pattern we saw in about 70% of the 50 sites. Some of them stayed about the same, and only in a couple conditions did they shift to a wetter condition, and I'll show you an example there. Here's another site that we evaluated in downtown Redmond. And this is the same bar chart showing the original plant list with categories in the blue bars, and what we see today shifting towards more in the facultative classification 10 years later. One of the 2 or 3 exceptions to this rule was Blodale Donovan Park in Bellingham. And this facility is located at the downstream end of a fairly large parking lot that serves also as a boat launch to Lake Whatcom, which you don't see off the screen here, about 200 feet away to the east. And the shore is actually physically uphill, perched, and apparently provides a groundwater source to this facility downstream, downhill. And we saw this through the original planting plan, again, blue, where they maybe anticipated it should be a little drier, and they put more dry plants in the initial planting plan. But now today we saw that it's shifted to wetter plants. And when Jenny and her group did hand borings of the soil, they did reach groundwater, as I recall. So this is one of the very few exceptions. In general, the sites had been planted with wetter end plants and really moved towards drier. And here's that conclusion as a summary across all the sites, that as a percent of the plant lists, they were largely wetter plants and later have shifted to drier plants. I think that that just represents a bit of a misconception. Hello? A bit of a misconception that somehow these would be very wet sites, and they're very much not. Summary of vegetation changes. A couple things to point out, that in the original plant list, there were about 3.1 herbs specified in the design. And in the observed plant list, what we see today, there are many, many more. And it looks like they're really mostly volunteers that are adding to that list of herbaceous plants, because the percent of the original plant list for the herbaceous plants was only 13.4%. So this is really showing maybe there's a little bit of more finesse that can be gained in identifying which plants to be specifying in these facilities. The woody plants, trees and shrubs, sustain themselves fairly better, and 56% of those remained as observed in the sites. One question might be, well, whether these sites, the plant WIS was reflecting the infiltration conditions. So maybe sites that had lower infiltration rates would support wetter plants, and plants that had, and sites that had higher infiltration rates would tend to support drier plants. So we looked at this and we separated this group by, as you can see as the header in each of the graphs, either typical design or underdrained. And typical just means that there's not an underdrain and the water flows through the bioretention facility and reaches the native subgrade beneath and infiltrates down through there. With underdrain facilities, the subgrade has such a constrained infiltration rate that it would accumulate too much water. And so the engineers, as you all have probably done, specify an underdrain that allows to drain off the infiltrated water to a conventional storm drain. And in the case of the typical, we did see a little bit of a pattern, very slight, except that there are 4 sites that are at the top up here that the subgrade was highly infiltrating, and that was seen in the geologic setting that the geologist identified. So this is pretty consistent with what was seen. With the underdrain facilities, this represents more the infiltration rate of just the bioretention media, and we don't see much relationship there. We also did a survey of the operations and maintenance supervisors for all of these owners, and they answered questions, a number of questions regarding what their activity is, whether there's irrigation, whether they replanted, did they replace the soil, what have you. This was one of the basics that I wanted to summarize for you all, that the frequency of maintenance was largely seasonal. That frequency amongst the 24 respondents or so of 1 to 4 times a year was the largest group, the 15, and many of those were even just 1 or 2 times a year, very seasonal. There was one that was much less frequent, and one that was never at all. They expected the facility to maintain itself, and they were not going to conduct maintenance activity. The other group that is a little bit more frequent, at 1 to 2 times per month, are sites where the aesthetic quality of the facilities was expected to be maintained for public appearance, and that was the main motivation for that frequency. The vegetation take-home points then were that for the particular sites, to anticipate which plants are going to survive well, you have to be aware of the unique site conditions. What's the moisture regime? What's the solar exposure? How dry is it going to be? Is there groundwater? Will there be irrigation? And where's the inflow location? What's the expected maintenance? Again, irrigation, how often? And what are the goals, the visual goals, never mind infiltration for water quality, for water control and water quality treatment? What are the aesthetic goals? Also, what's the contributing area ratio? Is there a large area contributing to the facility or less? Are there volunteers nearby? Is there habitat nearby that would contribute plants? And expect spatial variability within the site. Jenny will show an example of that in her work. And that woody vegetation persisted better than herbaceous. We also did a modeling evaluation of the designs that were conducted or that were provided, and we identified, and Doug Byerline of Clear Creek Solution looked through those. And one of the main things he looked at was what was the drainage, the cell area to drainage area ratio. Just how big are these bioretention cells relative to the drainage area contributing the water to them? And he identified a number of them that had very large drainage areas, or conversely said, very small cells relative to the drainage area. And the example in the first case, 0.1% means that the bioretention area was 1/1000th of the area of the contributing drainage basin. And these were all the very smallest cells with the largest drainage areas that he found. And even with that much hydraulic load to the facility, the infiltration rates were really quite high, ranging from 18 to over 100 inches per hour. So we will get into the soils and infiltration analysis with Jenny.
Hi, everybody. Jenny Saltonstall with Associated Earth Sciences. And so our portion, the geotechnical and hydrogeologic study for the bioretention cells, focused more on looking at the plans that were available. Reminder that all of these facilities were at least 10 years old. Some of them were 20 plus years old. And so step one for our work was trying to find the plans and then reviewing the plans and when available, any specification information, and then comparing that to what was on the ground. And so our workflow for that is we would attempt to get a drone image of the cell where the FAA would allow it, and that was our base map. And then, we went through the standard permissions, but what we really wanted to do was characterize the cell in a detailed fashion. So we had one long day in the field for our site assessment work, and that included shallow hand-augered boreholes to look at the bioretention soil thickness and what was underneath the bioretention soil. And we tried to get through the underdrained gravel if it was present to characterize the subgrade, that geologic unit underneath the bioretention soil. We installed a shallow well point so we could look at water on top of the soil during testing, and then what happened to that water as it passed through the bioretention soil. And then we used the geotechnical T-probe to look at compaction and collected samples for laboratory testing. Our lab testing was on the soil. This study did not do any water quality work. We were all about flow through the bioretention soil, through the bioretention cell. So the uppermost photo is one of those drone images, and the lower two photos on this slide show two different end members of vegetation cover in the cells during infiltration testing. And so the controlled infiltration test was modeled after, loosely after the pilot infiltration test methodology, but what we were attempting to do was a facility scale infiltration test. So when hydrants were available and the rates were not too high, we could get a pool of water across the facility up to the design ponding depth. That was our goal with our controlled infiltration test. Next slide, Bill. So the comparison with as-builts. So 3 cells we did not have as-builts for. So of the cells where we had as-builts, 38 compared pretty consistently with inlets, outlets, overflows. The cell seemed to behave per its design. And so what's always interesting when you're studying a large number of facilities are the outliers. And so for some outliers, for cells that varied in a substantial way from the as-builts, there were a number of variances, and we kind of grouped them into non-engineered overflows, uh, lateral bypass, and then a kind of a general category called landscaping modifications. So this upper picture, you see the round structure in the top of the top photo, that's the overflow. And then you can see the murky pool of water. We were able, in this controlled infiltration test, to bring the water across the facility, and we were trying to keep it just below the overflow. But for this cell, water began to flow out the side of the cell before it reached the overflow. And so that is a non-engineered overflow. And so for each one of these cells, we produced a site assessment report that'll go back to the facility owner so they can think about maintenance. Another example of bypasses in the lower photo, this was a small cell with a modular block wall on one side of it. And when we applied the water to the cell for the controlled test, when the water built up, it began to leak out the side there. Very just small amounts of seepage, but that's, obviously not ideal for a water pathway. And then landscaping modifications. There's kind of 2 end members to that. One is when a cell is filled in. So none of the cells selected for study were filled in, of course, but during site selection, we looked at many more cells than were selected. And we did have a couple of instances of a couple of cells just not being there. They looked like they had been filled in. The other end member is a cell that is very well loved by the adjacent residences, and maybe it's heavily maintained and maybe saw some compaction, or a homeowner may decide to fix a poorly draining cell and do some modifications. We saw both of those. Inlets and outlets, generally the inlets were in good shape. There was a little bit of erosion in 17 cells at an inlet. 32 cells had one or more inlet with blockage. And what we saw there was for cells with multiple inlets, pipes that maybe didn't bring a lot of flow in, sometimes were just buried with mulch or debris. And so not an effective inlet, didn't keep itself cleaned out. And then on the outlets, 41 cells had engineered overflows, and of those, some had some buildup of debris, usually organic matter, at the overflow. Next slide, Bill. So this is another example of a drone image for a bioretention cell, and this is what one of our site assessment maps looks like. And I want to walk you through this. It's a fairly large facility. And so on the right side of the map, there's an IN-1. That's the inlet where water comes into the cell. And then there is a skinny blue line with an arrow on the end of it that wiggles across the cell. That's the preferential flow path. And so we saw different versions of this in most of the cells. There was, over time, there's a microtopography that develops as water comes in one end, flows across the cell, maybe goes to the overflow or soaks in. And so along that blue flow path line, it's just a little bit lower than the adjacent cell base on either side. The light blue areas are where we conducted our controlled infiltration tests. And so the pink is our diffuser. We applied water through an energy dissipation device at a constant rate and then measured the wetted area. The cell had some pretty high infiltration rates, and so we were not able to fully wet the cell base. And so across that cell from the inlet, there's kind of an octagon shape. That is an area of standing water. It's right at an energy dissipation, and we have a near zero for that portion. And then in the flow path, we ran a infiltration test and measured about 40 inches an hour, a very good infiltration rate. Took a grab sample of the surface soils there to look at the fines content relative to what's a few inches below the surface. And then we conducted a second infiltration test outside of the flow path and had a higher infiltration rate and less fine-grained material. And so you can think about microtopography. If you were the manager of this facility, you might think about how you would maintain something like this, either to get rid of the flow path or to keep it, because generally the plants seem to be happier along this flow path because they had a little bit more fines. They saw water more frequently, much drier and better draining outside of the flow path. So a couple of different options there to think about. Next slide, Bill. Yeah, so 50 cells. And so trying to pull that information together and look for some common variables. So we looked at the geologic setting for the cells. For typical, and remember, typical bioretention is one that infiltrates out the bottom. There is no underdrained versus underdrained bioretention cells. So the plot on the left is some bar graphs showing the distribution of cells and geologic units. And so most of the typical bioretention cells are situated on geologic units that are considered permeable. So glacial outwash. Most of the typical cells were either on glacial outwash or alluvium. And then most of the underdrained bioretention cells were on glacial till or glacial drift, another fine-grained unit. There were a few cells where we couldn't tell what the bioretention cell was sitting in. We had a regional map, regional soils map, but they were either situated on rocky fill or we couldn't get through the underdrain with our hand augers to see what was underneath it. And so you can see that on that bar chart. And then the plot on the right gets at what Bill was talking about, and that is infiltration rates. And so just note the scale. We go from 0 to over 100 inches an hour. And so quite the orders of magnitude there in terms of infiltration rate. The left box and whiskers plot is for the typical bioretention cells, and you'll see that although they do have some very high infiltration rates, in general, the typical bioretention cells had a lower infiltration rate compared to the underdrained bioretention cells. The underdrained bioretention cells were generally had higher infiltration rates. And that's because what controls the infiltration rate? It's what's underneath the bioretention soil, if the soil's put in properly. And so the underdrain takes that water away, and so it's only the bioretention soil for an underdrained cell. But when it's an infiltrating cell, no underdrain, the infiltration rate is controlled by what's underneath the bioretention soil. Next
Looks like we have a
Bill.
question. Sorry, looks like we have a question just about infiltration rates really quick. How did the infiltration rates compare to the original infiltration rates before the retention facility was put in?
Yeah, that's a great question. Most of these sites did not have original field infiltration data from the time of construction available to us. And just based on my experience with how bioretention cells were built, there usually wasn't a construction phase infiltration test conducted. So that's a relatively new requirement to have that work done at the time of construction. Historically, there usually wasn't a construction phase infiltration test. What we did have was design rate information. And for some of the sites, we had the geotech report that talked about how they came up with the design infiltration rate. And in nearly all cases, this infiltration testing we did was higher than the design infiltration rates for the facilities. And so here we're talking about the bioretention soil itself. So that 18 inches of media above the underdrain drain or above the native subgrade. That is, an important component of the bioretention soil systems. And the plot on the left is comparing the infiltration rate during the facility infiltration test to the fines content of the bioretention soil. And so we collected samples of the bioretention soil and did grain size distribution tests and organic matter content testing on that material. And so we're comparing the fines content of the bioretention soil to the infiltration rate. And I've drawn 2 red circles on there, showing in general that when the fines content is less than about 10%, it was not a factor in how the infiltration rate in the facility scale infiltration rates, because we have a wide range of infiltration rates for a pretty consistent fines content. That just means there's other factors at play in performance of that infiltration facility. But as the fine content went up, the infiltration rate did go down. So there is, once you have enough fines in there, it does affect the infiltration capacity. Many folks were interested in how the bioretention soil media compares to the current default bioretention soil mix that's sometimes called the 60-40 mix. And so with the plot on the right, it's a grain size distribution chart, and there's a blue envelope there that shows the expected gradation for the default ecology bioretention soil mix. And most of the tested material was either within that envelope or was finer grained than the default 60-40 mix. Let's see. I think that mostly what I wanted to say about the bioretention soil bill. Next slide. Yeah, so our key findings here, I guess I want to preface this by saying, the field is messy. And so we as designers will put together a nice plan sheet that shows a beautiful bioretention soil, and what gets constructed and then maintained can look quite different over time. And so that microtopography or facility scale heterogeneity is really important to remember when coming back to look at a facility. And so I really like the controlled infiltration test, where we try to apply as much water as practical to the facility to get an average performance rate. If you do small scale testing, you may not get the full picture of how the facility performs during the bigger storm events, which is when we really want them to be working. And so really encourage use of when you can do it, the facility scale performance infiltration test for these bioretention cells. And usually if you have a hydrant, it's not too much trouble. And so key findings, it was good news. The study, we weren't sure what we would find with these very old bioretention facilities, but in general, they mostly resembled their original plans. And so I found that very encouraging, because we're only going to see the use of bioretention expand. Some key differences were those non-engineered overflows, lateral flow. Another type of lateral flow was a bioretention cell next to a pervious pavement subgrade. So this was a roadside bioretention setting. And when the water was ponded deep enough in the bioretention cell, it could laterally flow into the adjacent pervious pavement base course. So that was interesting to observe. And then leaky structures. So the overflow structures within the cells, those concrete sections, they almost all of them would leak at the joints. And so we'd be ponding water in the cell, and we'd hear water in the overflow before the water had hit the overflow elevation. And you look inside and you do have water coming in through those joints in the concrete sections. So when it's intended to be a fully infiltrating bioretention cell, having water go into the overflow instead of soaking into the ground is a bypass issue. And then, high infiltration rates persisted. So even facilities with low maintenance or kind of out of the way areas with variable sediment loading, we had, especially for the underdrained facilities, very high infiltration rates, even after a decade plus of time. Didn't see evidence of facility scale clogging. There was some sedimentation at inlets localized, but it didn't extend across the cells. We had a variety of land uses, schools, residential, arterial, and residential, or sorry, said residential twice. And so there was a good range of sediment inputs into the cells. And then the bioretention soil mix in general was finer than the current default mix, but still had good infiltration rates. And there was a wide range of organic content tests results.
And Jenny, one thing I brought up last time was your observation that there were some finer range of fine grained particles, but that those were probably from the original installation and not accumulation from inflow sediment load.
We targeted our soil sampling to be usually starting about 3 inches down. So we didn't include the mulch and we didn't include any surface scum when we were trying to characterize the bioretention soil. We did take some separate targeted samples to look at fines accumulation, but that wasn't part of the numbers for bioretention soil. Good clarification.
Yeah, good point. Okay. I think we're ready for questions, and I will discontinue sharing unless we need to go back to one of the graphs. And this is our favorite part, to hear questions from you all. Yes, Blake.
Hi, Bill and Jenny. Question for you. The ecology manual requires us to design bioretention cells facilities using 12 inches per hour, and then to add a KSAT safety factor depending on the contributing area. What are based on your findings, do you believe that to still be good based on your research? Do you think it should be higher? Do you think it should be lower? I'm curious to know what your thoughts are compared to what we actually have to design with.
Yeah, it's a good question. Bill, I'll jump in if that's okay.
Yes.
Yeah, it's a good question because we found pretty high infiltration rates generally. Many of them were above the 12 inches an hour. And so we've shared this with ecology. I'll just remind everybody that bioretention works great if it's installed correctly, but it's really easy for a low bid contractor to install it incorrectly at the wrong time of the year or with compactive equipment or just walking on it when it's wet, and you can quickly go from 12 to something lower than 12. And so I think ecology is just very conservative, with that in mind. But it's more of a policy question on how conservative ecology wants to be with design of the bioretention systems. I know that one of the interests in this study was how are they performing in time? They're supposed to have a facility life of, well, you guys know, several decades of life in terms of performance. And so that's, embedded in those initial rates and safety factors.
Thank you.
Nope. Yeah, no clear response there, right? Yeah.
That's okay. I understand. Thank you.
Yes, next. Lisa, is it next?
Sure. So I have questions about the plants.
Okay.
It looked like some of your pictures still had plants in them, but a lot of it looked like it was grass. And I know for a while there, the city of Kirkland was saying, give up on the plants, just go with grass because nobody's maintaining them and they look terrible. I don't know if that's changed or not. But were you finding anything where ones that had grass were doing okay as opposed to ones that had the deeper rooted plants?
So I'm speaking a little bit as I'm a more water quality specialist, so I'm speaking on behalf of our botanists that are absent today. But You're exactly right that there can be, well, as you saw, there was a transition of plants from what was originally planted to what's there now. And that transition can be as a result of a number of things. And just benign neglect, I think, will, with lack of maintenance and lack of irrigation, will support any shift that might be that they may tend to do if there's other nearby plants that are invasives, or the maintenance activity just becomes fairly aggressive. For example, there are a number of cells that we had near the Arlington airport, and some of the management requirement was to keep that vegetation low for purposes of safety of the airport. Also, there's a number of the maintenance folks said, hey, when we've got sight distances in traffic lanes, we have to specify plants that stay small. And they don't want to have to go in and keep trimming them down just for traffic sight distance reasons. There were a number of other sites that had specified seed mixes. One of the photos that we were looking at was one of those, and on the end of the seed mixes or other weedy species that might come in there, and there were some of the sites actually that were converted into lawn, and they were physically part of a neighbor's front yard, and they were running the mower down there, and there was no point at which where you could see their lawn transitioned into a bioretention facility. It was all contiguous turf. And what the infiltration rate literature is talking about is that the turf grasses form very dense, shallower root masses that can inhibit the infiltration. More prairie-like grasses actually have a very thin and branching root, taproot that can support infiltration. And then shrubs have a larger root, and trees, of course, even larger. And in each of those cases, there's greater infiltration supported by the vegetation. It's a little unclear to what extent the plant roots are really sustaining any infiltration, as Jenny reminds our team that this soil is very high infiltrating sandy soil, and whether the plant growth makes a difference there, it's a little unclear. It may be so sandy and draining that it limits the range of plants that you can select from, because some can't live in that dry of condition, which, as you saw in one of the photos from uh, alongside the road, major highway in Arlington, it just turns to weedy species that are mowed by maintenance people and probably gets walked over. So the planting strategy really should anticipate what the purpose of the plant community is and what the expected maintenance is going to be.
Thank you.
I see more hands. Paula.
Paula, you are on mute if you're trying to talk.
Oh, okay. Now I'm sorry about that. Yes, this is also a question for Bill about the plants, and you were actually talking about the herbation vegetation, and that's one of the things I didn't quite follow in your slideshow as far as what you were saying. Did the amount of herbaceous vegetation increase over time, or was it? And did you find that if there was an increase, it was just more weeds? Or I guess I am curious about, yeah, the herbaceous vegetation. What was the change? Was it a transition to just a more of a weed mix?
In many cases, yes. So that transition essentially showed that the number of herbaceous plants went up, the number of species, different species went up over time. And that most of those that increased in the number of species were not specified. They were not originally planted, so they were invasive from some source, either adjacent habitats or bird feces, what have you. And that amongst those, that increase of the herbaceous plants, only, so I'm trying to recall the exact number, only 13% were herbaceous species that were specified in the original planting plan. So not only did they increase, but there were not many of the original plants carrying over. I hope that clarifies things.
That did. Thank you.
We also have a couple of questions in the chat. Any thoughts on pollutant removal effectiveness given the high infiltration rates? Is that a part of the study at all?
Well, I can reply to that. One of the things that I really enjoy is reading a lot of the scientific literature, and I did so for a lot of this background material. A lot of that did address the water quality treatment, and it gets very complicated. There's a lot of interaction between, of course, the soil and the plant roots and the contact time and what have you. So we really are avoiding that topic and just focusing on whether the water is getting into the ground. There is some question about if there's such rapid infiltration, is there not enough contact time? So a lot of the papers talked about that trade-off between high infiltration rates to ensure that infiltration is sustained, and yet maybe bypassing a lot of the contact time. There were a couple of sites, for instance, in the previous study and in this study, where the infiltration rates were so high that Jenny had to just make small pools to try to measure the infiltration rate, which would suggest that even with some of the moderate or some size of larger storms, that as the water entered, it's not spreading out across the bioretention facility. We had a site, I believe we actually tested it again in this study, that pretty much within a fourth of the area of the facility with fairly large storms, it was immediately bypassing through the soil and into the underdrain. And 3/4 of the facility was not getting any contact time with any of the water. So it turns into a finesse between that infiltration rate and, if you're talking about water quality, whether it's getting enough contact time to have that water quality removal process. I hope that answered your question. Jenny, do you have any additional comment on that?
Well, water quality treatment for the different types of bioretention soil mixes was done at the WSU Puyallup Extension Station there with some column studies when they were developing the default 60-40 mix. And so all of those columns had high infiltration rates, in excess of 20 inches an hour. And they did water quality testing of artificially contaminated water to look at effectiveness. And so there are some papers on that. And so, it was part of coming up with the 60-40 mix, 60% sand, 40% compost for the default mix. Good question.
All right, and then the last question that we have is if there's a list of plants available that the study found that are most successful in the facilities. And then the second question, or part 2, is there a report available for the study?
A report is forthcoming, and the I think we were asked this question about a plant list of the most successful plants in each of our presentations. And really what it comes down to is that for preparing a site planting plan for facilities, you want to look at the site specific conditions very carefully. Just how hot does it get? What's the aspect? Is there shade? Is it up against a building adjacent to hot parking lot or freeway? Is it in a cooler environment? Is it on top of, if it's not a under drain facility, if it's typical? Is it on top of highly draining subgrades that will effectively drain away the water quite quickly? And also, maybe most importantly, what are the aesthetic goals and the level of maintenance and the budget for maintenance in a capital management sense? What's the plan for the facility? Because if you're specifying a plant palette that is a number of expensive plants that takes a lot of maintenance for some kind of aesthetic purpose, but you don't have the budget to maintain it, there's a discontinuity there. So it takes a lot of site-specific and organizational awareness for the site to do your plant selection. I thought that, for example, the 2 Whatcom County sites that were specified with a seed mix seemed to be a reasonable planting plan. They maintain it with a line trimmer twice a year and rake up some of the cut vegetation and call it good. The Skagit County Public Works Building has a very attractive bioretention facility that gets irrigated and well maintained, and maybe some periodic replanting. So being very aware of the site conditions, both the environmental site conditions and the organizational goals for the site is what guides this plant selection.
Awesome. Thank you. Are there any last minute questions for our presenters? All right. Well, I think you were very thorough in your presentation then, and thank you so much for putting in the time to meet with us all today. And I guess with that, that'll be a wrap for this hour. Thank you so much for attending. And yeah, again, thank you so much, Bill and Jenny, for presenting.
Thank you all.
Of course.
Thank you.
Thanks, everybody.
Giovanna and Caitlin, do you need to